cover some basics and everything on a 2.2 cell structure as part of cells as a topic. So to start off with, cell theory as proposed back when microscopes were first discovered. And the first one we have are cells are the smallest unit of life. And this one's pretty self-explanatory. Everything is built up in different systems, but everything traces back to cells. Second one is cells arise from pre-existing cells. Things can't just come out of nowhere. They have to evolve and adapt into different things. And the third one is all living organisms are made of one or more cells. And our levels of organization we're looking at here are cells that form tissues, that form organs, that form organ systems, that make up an organism. And our key concept is unicellular organisms have one cell, which is the uni, and multicellular organisms have many cells. And then there are 4 kingdoms of life. And we're usually more familiar with these 3, fungi, plants, animals, but we have protista, which they're eukaryotic cells, but you don't really see them very often. They're like amoeba. Let's move into a quick overview on prokaryotic versus eukaryotic cells. So if I just scroll over here really quick, we have, this is a prokaryotic cell, and then everything on the rest of the page are eukaryotic cells. So to start off, a prokaryotic cell, they're mostly bacteria, is what we're looking at. The IB syllabus. And prokaryotic cells have naked DNA, so they have no histone proteins, which are these proteins that bind and coil DNA around, so their DNA just kind of scattered. They have circular DNA, and their DNA is a closed loop, and they have a cell wall made of peptidoglycan, and they reproduce via bacterial conjugation. So they basically do the bacterial version of mitosis, and they split. Whereas in eukaryotic cells, they are complex structures and they have membrane-bound organelles, so things within the eukaryotic cell itself. And they can be either uni or multicellular organisms. And these are the key things that you would typically find within a eukaryotic cell. So we'll go over, look at the cell structures first and everything inside. To start off with the prokaryotic cell structure, again, we have the capsule, which is made up of the cell wall, and it's peptidoglycan material, and it prevents lysis, which is basically the splitting of the cell. And on the outside, we also have these pili, which are attachments to aid in bacterial conjugation, which is how they reproduce. And then we have the flagellum for propulsion and movement around the environment that it's surviving in. And then they also have a plasma membrane, so there's really just a lot of layers to the prokaryotic cell, but they have a plasma membrane. And then within that, we have plasmid and nucleoDNA. And nucleoDNA is really just non-membrane bounded naked DNA with no histone proteins attaching it together. And then we have 70S ribosomes, and this is important because eukaryotic cells have 80S. And one of the main distinguishments between prokaryotic cells and eukaryotic cells are their ribosome number. So prokaryotic cells have 70S ribosomes, and they have cytoplasm, which is the internal fluid that all of these things reside within. And then if we look at our typical eukaryotic cell structure, and usually we illustrate this with an animal cell diagram, but it's not completely necessary. We have cytoplasm. Animal cells don't have a cell wall, but they have a cell membrane. And then we have free-moving ribosomes, and they're 80S, which is the main distinguishment because in prokaryotic cells they were 70S, in eukaryotic cells they're 80S. 80S ribosomes, if they are not bound to anything and they're free-moving, then they synthesize proteins for use within the cell. We have peroxisomes for detox purposes. And then this is the rough endoplasmic reticulum, and we see the ribosomes are embedded on the outside of the rough ER, and because we know ribosomes are a site of protein synthesis, the rough ER is where proteins are synthesized for use outside the cell. And then we have cilia on the outside of cells to aid in movement. And they're not as powerful as the flagellum were, but they do help move the cell around a fair bit. And then mitochondrion, which generate cellular energy through cellular respiration, vacuoles for storage and waste removal, and then centrioles, which are really key to nuclear division and mitosis. Then we have the smooth ER, which is the same as the rough ER, except it has no ribosomes studded upon it, and it's the synthesis of lipids and phospholipids. Then obviously we have the nucleus, which is surrounded by the nuclear envelope, and this allows for selective expression of genes, which are really critical for transcription. And then we have the Golgi apparatus, which helps store and transport and packs proteins for exocytosis out of the cell. And then lysosomes are kind of just containers to transport enzymes around. Yeah. But for this subtopic, you don't really need to know in depth you need to know what the processes are, but not in depth how the processes work. Some differentiations between the eukaryotic cells are the plant cell structure. So plant cells have a cell wall, and this helps keep the plant cell nice and compact within a structural shape. They have a larger vacuole for structural support. And then again, they have ADS ribosomes, and everything else remains the same. And they have chloroplasts, which aid in photosynthesis. And then for a fungal cell structure, they're a lot rounder, but they do have a cell wall, and it's made of chitin, and they have budding scars, which is how they reproduce. So they go through reproduction, and they split, and they produce a new bud, which then replicates the inside of it, and then on the original cell, they end up with a budding scar. And so however many budding scars a fungal cell has is how many times they've reproduced. Yeah, and that is everything for the typical cell structure. Yeah. And then to look at some atypical cell structures, which are just really abnormal features that are not commonly found within normal cells. There are a few, and the main four ones we need to know for the IB syllabus is aseptate fungi, which are multinucleated, and they lack a divide in the hyphae, which means they don't have the ending plate in the middle, so each cell kind of combines with the next one, and so they're considered as multinucleate. And then skeletal muscle cells, because they're large in size and they branch through your body, and they're also multinucleate because they need to generate lots of energy and to provide a basis for all the metabolic reactions that happen within your body. And then we have red blood cells, also known as erythrocytes, which are bioconcave disc-shaped like structures, and they're anucleate because you need to maximize your surface area to volume ratio of the cell. And then we have sieve tube elements, which are found in plants, and they're also anucleate because they really just aid in transportation purposes, though there's no point of taking up extra space with a nucleus. And then when we want to study cells, usually we put them under microscopes, but for some really minuscule samples, you're not really able to see them properly, so we need to stain the sample. And for bacterial cell types, what mainly differentiates the two types of bacterium that we see is that they're either gram-positive or gram-negative, and when we stain them with a crystal violet stain, the ones that are gram-positive with no outer membrane retains that stain. But some types of bacteria, they also have structural differences, and so they may have an outer membrane. And if there is an outer membrane, then they are gram-negative, and they decolorize the stain because it takes longer for the stain to go through the membrane, and it diffuses and it loses color. Yeah.